Today we are going to cover this chapter, Respiration in Plants. So keep your NCERT with you and just underline all the important point which I am covering line by line from NCERT.
First, it starts with the energy, which is the source of light, and it says that energy of life process come from oxidation of macromolecule, which is obtained from food. Then it says that autotrophic nutrition, basically green plant and cyanobacteria prepare their own food by photosynthesis. Here protista is not mentioned. Remember, protista also perform photosynthesis. These autotrophic organisms trap the light energy and convert it into chemical energy and store it in the form of carbohydrate, that is glucose, sucrose and starch. In plants, they can have chloroplast containing cell, which is the green cell, and non-green tissue. Now non-green tissue will not perform photosynthesis, so they will depend on translocated food which will come from the green part of the plant. Heterotrophic nutrition, they are unable to produce their own food. Animal, fungi and saprophytes will come under heterotrophs, and they may be directly dependent on plant or they may be indirectly dependent on plant for food. So ultimately, all the living organisms depend on photosynthesis for food, whether it is directly or indirectly. Inside the cell, the cellular respiration takes place, and here the cellular respiration takes place in the cytoplasm and mitochondria. In prokaryote, only cytoplasm. In eukaryote, both cytoplasm and mitochondria. Now, in this cellular respiration, basically the carbon-carbon bond of the respiratory substrate is broken, and in that process, energy is released. If we talk about respiratory substrate, then these four molecules, carbohydrate, protein, fat and organic acid, can act as a respiratory substrate. Out of these four, carbohydrate is the primary respiratory substrate, and among carbohydrate, glucose is the most common respiratory substrate. Now, this respiratory substrate release energy not all at once, but it is a slow process, and the energy is released in a step-by-step process. Each step is controlled by an enzyme. This ensures that there is no energy wastage and allow a very efficient use of energy. Now, the energy released due to breakdown of these respiratory substrate is used to synthesize ATP, adenosine triphosphate, which is an energy currency. This ATP is stored energy and it releases energy as required by in the cellular function. The synthesis of carbon skeleton in biosynthesis of several molecules require energy, so here ATP is used. Now, plant require oxygen. Plant require oxygen for respiration, so basically they consume oxygen and they release CO2. Plant do not have any specialized respiratory organ like animals, but they do have stomata, which is present usually in the leaves, and lenticels, which is present mainly in the stem, for gaseous exchange. Then the question arises, why plant can survive without respiratory organ? So there are several points given in that paragraph. Because each part of the plant manage their own gaseous exchange. Leaf will do their own exchange, root will do their own exchange, stem will do their own exchange. Then the rate of respiration of plant is very low as compared to animal, and moreover plant also do photosynthesis, which will release oxygen, and most of the oxygen demand of respiration is fulfilled by the photosynthesis. These gas which are exchanged in the plants, they have a very short diffusing distance. Anyway, diffusion of gas has a very short distance in plant. The woody stem and the root which is present, they have lenticels for gaseous exchange. Leaf have stomata, stem have lenticels. Now these part of the plant, they have loose packing of parenchymatous cell which have intercellular space, and these will have large air space. Most cell of the plant have at least a part of their surface in contact with air. In plant, glucose as the respiratory substrate will break down completely into CO2, H2O and energy. This breakdown is a multi-step process in which there is a stepwise oxidation of substrate which will allow ATP synthesis. So the earlier point was for the aerobic respiration. Now if we see anaerobic respiration, then cells can survive without oxygen also in anaerobic condition. In fact, the earliest life form were used to survive in an oxygen-free atmosphere. So we can say that the earliest life form were anaerobic in nature. Anaerobic organism even exist today, and here two important terms are given. One is called facultative anaerobe, means who can survive with or without oxygen, and obligate anaerobe. Obligate means they can survive only in a fixed given condition. Now, if we talk about glycolysis, so it is made up of two words, glycose and lysis means splitting of sugar. This is also called as EMP pathway on the name of three scientists, Emden, Meyerhoff and Parnas, who discovered this pathway. Now, it is the only respiratory process in anaerobic organism, because in anaerobic organism the ATP produced is actually produced in the glycolysis. We will see it later. If we talk about the location of this glycolysis, so it takes place in the cytoplasm and it takes place in all the living organism, whether it is aerobic or anaerobic. It is common pathway between both aerobic and anaerobic organism. Now here there is a partial oxidation of glucose. As we have seen that a complete oxidation always produce CO2 and H2O. But here, since two molecule of pyruvic acid is produced, which is 3 carbon molecule, so that is why it is a partial oxidation of glucose. Glucose is derived from sucrose, which is a disaccharide, and it is digested by an enzyme invertase, which convert one molecule of sucrose into glucose and fructose, and these glucose and fructose both can enter into the glycolysis. Moving on to the steps of glycolysis, so as you can see here that glycolysis is a 10-step reaction. Now these 10-step reaction are divided into several phases. So the first phase is the phosphorylation phase, in which glucose is converted into glucose 6-phosphate. Mind it, here the enzyme involved is hexokinase. Since here a phosphate is added to glucose, so here ATP will be used. So phosphate coming is from the ATP. Then this glucose 6-phosphate is converted into fructose 6-phosphate. This is a isomerization reaction. So here enzyme isomerase will be used. Then this fructose 6-phosphate will again add one more phosphate and it becomes fructose 1,6 bisphosphate. So here, since one more phosphate is again used, here ATP will be used. So here we can see that these phosphorylation step, ATP for in the first step and ATP is used in the third step. Now here this fructose 6-phosphate to fructose 1,6 bisphosphate require an enzyme phosphofructokinase. Now here also note one thing, that if here glucose is entering into the into the pathway, if fructose enter into the pathway, then fructose will directly get converted into fructose 6-phosphate, and here the isomerization step do not take place. Now here conversion of fructose to fructose 6-phosphate, this is also catalyzed by the enzyme hexokinase. Now once this fructose 1,6 bisphosphate is formed, then the pathway will enter into the splitting phase. And in this splitting phase, what happens, as you can see here, the 6 carbon fructose 1,6 bisphosphate get converted into 2,3 carbon molecule. One is called the PGAL, or also called as glyceraldehyde 3-phosphate, as you can see here in the pathway, and DHAP, dihydroxyacetone phosphate. Now this dihydroxyacetone phosphate ultimately get converted into PGAL, and two molecule of PGAL will further continue into the cycle. So after the splitting phase, whatever is formed in the pathway will be multiplied by 2, as you can see here in the pathway also. Now this PGAL, which has one phosphate, here one more phosphate is added, and this PGAL become 1,3 biphosphoglycerate, also called as BPGA. Now here if you see, in this step, PGAL to 1,3 BPGA. Here one phosphate is added, but from where this phosphate is coming? So here phosphate is added, but it is not coming from ATP. It is coming from inorganic phosphate. Now mind it here that one NADH is released. So the first ATP will be synthesized when BPGA lose its one phosphate and become 3-PGA, 3-phosphoglycerate. So here one ATP will be formed. Then this 3-phosphoglycerate will convert into 2-phosphoglycerate. This is an isomerization step and no ATP is formed here. Then this 2-phosphoglycerate will get converted into phosphoenol pyruvate. Here one H2O will be released, but in this step there is no ATP released. Then this phosphoenol pyruvate will get converted into pyruvic acid. So here again we can see that one phosphate is released and that phosphate get trapped into ATP. So here in the ATP synthesis phase we can clearly see that 2 ATP is produced. Now overall if we see after the splitting phase everything is multiplied by 2. So NADH is produced, so 2 NADH will be produced and these ATP will be multiplied by 2, so 4 ATP is produced. Now also note that earlier in the beginning 2 ATP was used. So whenever we will calculate the net ATP, 2 ATP we will subtract. So if we proceed further and see the ATP produced during the glycolysis, so ATP used is 2 molecule, ATP produced is 4 molecule. So the net gain of ATP 4 minus 2, that is 2 ATP is produced. In addition to that there is 2 NADH is also formed. Glycolysis produced 2 molecule of pyruvic acid which is 3 carbon, 2 NADH and 2 net ATP. So the pyruvic acid which is produced during glycolysis can have 3 fates. Either they can undergo lactic acid fermentation or alcoholic fermentation, or they can do aerobic respiration. The first two, that is the lactic acid fermentation and alcoholic fermentation, are part of anaerobic respiration. In aerobic respiration, as I said earlier, that now the mitochondria will get involved. This fermentation overall, it is an incomplete oxidation of glucose, because we will see that in the end CO2 and H2O is not produced only. Here ethanol is produced in alcoholic fermentation which is 2 carbon, and lactic acid is produced which is 3 carbon. So it is clearly a incomplete oxidation of glucose. Alcoholic fermentation take place in yeast, and here the pyruvic acid get converted into ethanol which is 2 carbon and CO2. So here one CO2 is released, but there is additional ethanol, that is why it is a incomplete oxidation. Now in this process pyruvate decarboxylase and alcoholic dehydrogenase, these are the two enzymes which are involved. Now mind it, this pyruvic acid to ethanol and CO2 production, it is a two-step process which may be represented here in 2 arrows also. Actually first the pyruvic acid is converted into acetaldehyde, and that process required this enzyme pyruvate decarboxylase, and acetaldehyde is 2 carbon. So in first step of alcoholic fermentation, pyruvic acid is converted into acetaldehyde plus CO2, and that is catalyzed by pyruvate decarboxylase. And in the second step, the acetaldehyde is converted into ethanol, which is catalyzed by the alcohol dehydrogenase. Now this in the lactic acid fermentation, which take place in mussels and bacteria, here the pyruvic acid which is 3 carbon get converted into 3 carbon lactic acid. Here the enzyme involved is lactate dehydrogenase. Here the enzymes are important, so keep these enzymes in mind. Now here if you see in this diagram, in both the alcoholic fermentation and lactic acid fermentation, NADH is consumed. See, NADH is produced in glycolysis, and this produced NADH is consumed by pyruvic acid into the lactic acid fermentation or the alcoholic fermentation. Any cell, either it can perform lactic acid fermentation or it can perform alcoholic fermentation. Same cell cannot perform both the type of fermentation. NADH produced is consumed in this fermentation step. So what is the net product of fermentation? So only 2 ATP is produced per molecule of glucose, and that also these 2 ATP is produced in the glycolysis step. Acid or these ethanol, in both the case, even the lactic acid or the ethanol, if they start accumulating in the cell, after a point they can become toxic. And one of the example given in NCERT is regarding the yeast, in which naturally occurring alcohol can have maximum of 13% alcohol. If you want to increase the alcohol content, then in that case the process of distillation is done. The third fate of pyruvic acid is aerobic respiration. So now the main character of aerobic respiration is that the substrate, that is glucose, is finally break down into CO2 and H2O. So this is called the complete oxidation of glucose. This aerobic respiration require mitochondria also. So the glycolysis part take place in cytoplasm, and then the aerobic step will take place in the mitochondria. Here, two crucial events are: first point, that all the carbon bond present in the pyruvic acid will get converted into CO2. Means all the carbon-carbon bond will be broken. And second, the NADH and FADH which is formed during this process will be converted into ATP with the help of ATP synthase enzyme, and here oxygen will also have a role. For that, the pyruvate which is present in the cytoplasm, this pyruvate will enter first into the mitochondria. Inside the mitochondria, this pyruvic acid will get converted into acetyl coenzyme A, carbon dioxide, and NADH is released. Now see here, this acetyl coenzyme A is 2 carbon and 1 CO2 is released. This process is called oxidative decarboxylation. So because here CO2 is released, so decarboxylation. Now in this process, enzyme is involved, and the name of enzyme is pyruvate dehydrogenase enzyme. In this step of oxidative decarboxylation, which is also sometimes called as link reaction, will produce 2 NADH in total. Now here one more thing to note is that Mg2+. In the functioning of pyruvate dehydrogenase, this Mg2+ play a very important role. The acetyl coenzyme A enters into the TCA cycle, also called the citric acid cycle. Now here, the acetyl coenzyme A reacts with 4-carbon oxaloacetic acid, and this 2-carbon plus 4-carbon form a citric acid which is of 6 carbon. This process is catalyzed by the enzyme citrate synthase. Now in the next step, that is called the isomerization step, the citrate is converted into isocitrate. Isomerization step, citrate become isocitrate. Now then it is followed by 2 decarboxylation step, where the isocitrate get converted into alpha ketoglutarate. So isocitrate is 6 carbon, alpha ketoglutarate 5 carbon, so there will be a loss of CO2 here. Now the alpha ketoglutarate, which is 5 carbon, get converted into a 4 carbon succinyl coenzyme A, so there is a loss of CO2. So here we can see in 2 subsequent steps there is release of 2 CO2. That is why these 2 steps are called the decarboxylation step. Here also note that in both the steps, 1 NADH each is produced in both the steps. Then this succinyl coenzyme A get converted into succinic acid, and in this process 1 GTP is produced. Now here this GTP is converted into ATP via a coupled reaction. Basically first GTP is formed, then GTP breakdown, and in place of GTP, ATP is formed. So in simple term, you can keep it in mind that here 1 ATP is produced. Succinic acid get converted into malic acid. That is, succinate get converted into malate, and then malate get converted into oxaloacetic acid. So here conversion of succinate into malate release another FADH2, and conversion of malate into oxaloacetic acid release NADH. So overall, if we see the entire cycle in one citric acid cycle, we can clearly see that there is a formation of 3 NADH, there is formation of 1 FADH, and there is formation of 1 ATP, which is from GTP. So here one line is given in NCERT for the cycle continuation, that is, succinic acid is oxidized, eventually regenerating oxaloacetic acid, and continuous oxidation of acetyl coenzyme A require replenishment of oxaloacetic acid and regeneration of NAD plus and FAD plus from NADH and FADH. So overall in the mitochondria we can see 3 CO2 is released, means pyruvate which has 3 carbon, now it is converted into 3 CO2. Along with 3 CO2, 4 NADH is produced and 1 FADH is produced, 1 ATP is produced. Now all this will be multiplied by 2 because 2 pyruvate is produced. So overall what we observe here, that 8 NADH, 2 FADH and 2 ATP is produced. Now the NADH and FADH produced during this process, now that need to be converted into ATP, and for that we need to go through the electron transport system. The electron transport system will take place in the inner mitochondrial membrane. Now here if you can see, there are 5 complex present here. As you can see, 1, then 3, 2, 4, and last one we also called as complex 5. NADH will come to complex 1 and FADH will come to complex 2. Now here NADH will convert into NAD plus, and in that process 2 electron is released. Now you can see clearly that these 2 electron through FMN through FES will go to UQ, that is ubiquinone. Then in this complex number 2 also the same thing will happen. FADH will break down. It will release 2 electron and these 2 electron will go to ubiquinone. So from both sides the electron goes to ubiquinone. Now this ubiquinone transfer this electron to complex 3. So from both sides the electron is transferred, and then through cytochrome B, FES and C, this electron will go to cytochrome C. From cytochrome C, as you can see here, cytochrome C is also called the mobile electron carrier because you can see it is moving between complex 3 and complex 4. Now cytochrome C transfer the electron to copper, which transfer it to cytochrome A, A3, and finally another copper, and it is then finally transferred to oxygen. So oxygen is the final electron acceptor, and this oxygen after accepting the electron become H2O. Now here there are a few things to observe very carefully. So let's see that. First of all, here you can see H plus is crossing the complex 1. So this we can call it as proton pump. Then you can also see complex 3, H plus is entering inside. In complex 4 also H plus is entering inside. So, but in complex 2 if you see, there is no H plus entering inside complex 2. So complex 2 you can say it is impermeable to H plus, whereas complex 1, 3, 4 and 5 are permeable to H plus. Now if you see in complex 1, 3 and 4, the H plus is going from matrix to intermembrane space, and it is accumulating in the intermembrane space. So H plus concentration will be very high in the intermembrane space, and then this H plus will want to go outside toward the matrix. Now this membrane is not permeable to the H plus. So here ATP synthase will come to the rescue, and here ATP synthase will provide a channel. So here you can see there is F0 and F1 particle. F0 is embedded in the membrane, F1 is protruding toward the matrix. So F1 act as a channel and F1 convert the ADP into ATP. Now here remember 4H plus will cross the membrane and it will lead to formation of 1 ATP. In old NCERT it is given as 2H plus. So don't remember that, right? Remember the latest which is given in NCERT, that 4H plus is present and this 4H plus will produce 1 ATP when it passes through the ATP synthase. In ETS we observe that the NADH and FADH, which is providing electron, is getting converted into ATP. Here 1 NADH will produce 3 ATP, 1 FADH is producing 2 ATP. If I summarize, there are total 38 ATP. Out of these 38 ATP, 4 ATP is substrate level. It is substrate level, and 34 ATP is the oxidative phosphorylation. Question is asked that from one molecule of glucose, how many ATP will be produced which is substrate level? So 4 ATP is produced substrate level, and from oxidative phosphorylation 34 ATP. Respiratory balance sheet assumption is that we consider that this is a sequential and orderly pathway in which the glycolysis will be followed by TCA cycle, will be followed by ETS, right? Means the product of glycolysis enter into TCA cycle and product of TCA cycle enter into ETS. So this is what is the basic assumption. Then we also assume that NADH from glycolysis enter into mitochondria. Then we also presume that there is no intermediate which is used in some other compound synthesis, and only glucose is respired without any alternative substrate. So these are some of the basic assumptions on which we have just calculated the 38 ATP. In reality, but what happened that The pathway works simultaneously, means in between it's not necessary that all the pyruvate will enter into the Krebs cycle. Some pyruvate will go in some other cycle also. Similarly, there can be certain additional substrate which can enter into the cycle. So different substrate can enter and exit from the cycle as needed by the cell. So in this process, ATP is used as it is required. So these activities are enzymatically controlled. So whatever this 38 ATP we have just calculated is actually a theoretical concept. Practically, it may be less also. Now, if we compare this aerobic respiration with fermentation, then we clearly see that in fermentation there is a partial breakdown of glucose. In aerobic respiration, there is a complete breakdown of glucose. In fermentation, there is only 2 ATP produced per molecule of glucose, whereas in aerobic respiration, 38 ATP is produced per molecule of glucose. Now, this NADH oxidation is slow in case of fermentation, but this NADH oxidation is vigorous in case of aerobic respiration. So overall, if we summarize the balance sheet, so as we have seen earlier also, that glycolysis will have 2 ATP plus 2 NADH. This NADH through ETS will produce 6 ATP. So total ATP in glycolysis is 8. So remember, in glycolysis 2 ATP is substrate level and 6 ATP is produced through oxidative phosphorylation. Then next step is the pyruvate oxidation, which will produce 2 NADH, also known as the link reaction or oxidative decarboxylation. So this step produces 6 ATP. Then Krebs cycle overall produce 6 NADH, 2 FADH and 2 ATP. So overall, if we see here, it produces 24 ATP. Out of this 24, you can say 2 ATP is the substrate level ATP, 22 ATP is the oxidative phosphorylation level. So overall, the grand total is 38 ATP per glucose. Now, as I said, that this is the theoretical thing. In real sense, it may be even lower because of certain loss of ATP. The respiratory pathway is an amphibolic pathway. It is considered amphibolic because it consists of both catabolic and anabolic process. Actually, in reality, there can be substrate which can enter into the respiratory pathway, and there are substrate which will exit also from the respiratory pathway. So here, some of the entry points is given. As you can see here, the fat which will break down into glycerol and fatty acid, and both glycerol and fatty acid can enter inside the respiratory pathway. Glycerol can directly enter into glycolysis, whereas fatty acid can get converted into acetyl coenzyme A, and then they can enter into the respiratory cycle through the Krebs cycle. Then protein, which break down into amino acid, and these amino acid can get converted into pyruvate, acetyl coenzyme A, or any other Krebs cycle intermediate and enter into the respiratory pathway. So we can clearly see that there are molecules which will enter into the respiratory pathway. In the same way, there are molecules which can exit from the respiratory pathway as well. For example, if cell need fatty acid, then acetyl coenzyme A, which is having a role in respiratory pathway, will be taken out from the respiratory pathway and fatty acid can be synthesized. On the other hand, Krebs cycle intermediates can also be taken out and they can be formed or converted into the amino acid. Hence, it makes the respiratory pathway a amphibolic pathway. Respiratory quotient is simply defined as the CO2 involved divided by the oxygen consumed. If the CO2 involved and oxygen consumed is equal, then the RQ will be 1, as in case of carbohydrate. Whereas in case of fat and protein, the RQ is less than 1. Fat, specifically it is 0.7 and protein it is 0.9. Lesser the RQ means more oxygen it is required for its oxidation. So organic acid can have more than respiratory quotient more than 1. Different respiratory substrate will have different respiratory quotient. That is it with this chapter. Now what you need to do is that take out your NCERT, read all these points line by line and memorize it. That is very important. So now please comment and let me know what is the next topic which you want me to cover. Also, if you want this PDF, then please comment your email ID and then there I will send you the PDF.